directly.
Hello, everyone. Welcome to session 4, Diseases. If you have been to my sessions at HCA Asia, you know they're always the best sessions. So welcome to the best session. It's an absolute privilege to be here, to be chairing this session, and also to be giving the first talk, the framing talk for this session on diseases. May I have my slides, please? It will be the best session once I have my slides. Wonderful. So I am Senjuti Saha. I work at the Child Health Research Foundation in Bangladesh, and I really work in the intersection of public health and microbes. We heard yesterday from Sarah, Aviv, Chloe that diseases must be part of the atlas going forward in the next 5 years. It has also been included in the strategic goals for HCA. I want to continue making that case that diseases must be a part of the human cell atlas because health is not universal state of humans. All human life, throughout our lifespan, we face challenges in health, so we must understand what are those changes in health. Also, diseases are common, diseases are complex, and diseases are inequitable. So again, reasons why we should study disease. Now, in the next 12 minutes or so I'll talk to you a little bit about the global burden of diseases. Now, what does burden look like? What the next 25 years is predicted to look like? What are some of the priority pathogens or priority diseases by the WHO at the moment and for the future? And of course, because I'm on stage, I'll talk a little bit about our work in Bangladesh on single cell genomics. So starting with the global burden of disease, Delhi. So I said in the intersection of public health and molecular biology, so I thought I would give you a little bit of public health primer or epidemiology primer. We talk about disease burden, but how do we measure burden? What does burden really mean? Is burden only the sheer number of deaths a disease can cause? In public health or epidemiology, we calculate burden using a term called the DALY. Higher the DALY, higher the burden. So what is DALY? So let's say this is the typical lifespan of a healthy human being. A child is born, a girl is born, let's say her name is Susanna, and she is expected to live a certain number of years. If it's in Singapore, she is expected to live about 83 years. However, let's say an accident happens. Unfortunately, Susanna dies and she loses a few years of life. That is called years of life lost, or YLL. We love abbreviations in epidemiology. Let's say before she died, she got into an accident, and because of that accident, she was in bed for many years. So she was not able to work, and due to illness, she was disabled, and she again lost, years there even before she died. And throughout her entire lifespan, she saw several diseases which kept her away from work, and she was disabled. These are years lived with disability, YLD. DALY is basically the summation of years lived with disability plus years of life lost. So that's really how we calculate global burden of disease. It's not only death. So you can imagine burden of a disease, even if it's very rare but kills children, has very high DALY, unlike some diseases that might kill us later in life, let's say some rare forms of cancer. So when we choose diseases, Sham talked about it today, it's very, very important that we understand the burden of the diseases we study, because our focus must be aligned with the global burden. And I'll tell you a little bit about what the burdens are of several diseases now. But in addition to that, Chloe talked about it yesterday, Sham talked about it, that burden cannot be the only thing. We of course have to ensure that we have access to samples, we have enough number of samples, these samples were equitably collected, they represent geography, they represent people, different diverse backgrounds, and of course as scientists, our currency is really our public publications and our interest and what we get out of it, right? So we must have passion and expertise in whatever we study. But the main point is for everybody, that circle is going to look different, but we really hope that there is some kind of an intersection of everything when we pick diseases to study. So what are the global diseases with the highest burden? So IHME is an institute that is commissioned by the Gates Foundation regularly to calculate all the diseases, to calculate burden of the most common diseases across the world. So this is the latest from 2022. This is the top 10 diseases in the world with the highest burden. On top is heart disease, followed by neonatal disorders, stroke, lower respiratory infections, etc. So it's really a combination of communicable diseases and non-communicable diseases, and also injuries. So you see that road injuries is actually in the top 10 because it does lead to early death, death in early life. Now, what does it look like in 2050? So IHME also does a lot of prediction, and using mathematical modeling, they can tell us what approximately, diseases will look like, or the top 10 diseases will look like in 2050. So the first thing you will notice over here that there isn't a lot of changes. A lot of the diseases right now also show up in 2050. For example, heart disease continues to be on top. Neonatal disorders come down a little bit, and there are a couple of diseases that show up in 2050 that we do not see in the top 10 diseases now. For example, chronic kidney disease or Alzheimer's disease. So this kind of tells us what we might want to focus on in the next 25 years or so. And at the same time, it gives, I think, HCA this opportunity to have an impact, to perhaps interrupt some of these trends that we do not like. I want to specifically talk a little more about neonatal disorders. So neonatal disorders, neonates are babies in their first 28 days of life. Believe it or not, 1 in every 20 babies born do not make it in their first month. And most of these deaths are occurring in low and middle income countries, so countries in the global south. This is something I'll talk about in a bit again. And the other disease, communicable disease, is lower respiratory infection, so caused by, let's say, influenza, respiratory syncytial virus, amongst others. Now, respiratory viruses are common all across the world, but severity and death are most common in countries with the least health resources. So 97% of all deaths due to pneumonia actually occur in just a few countries. COVID is also a lower respiratory tract infection, but it was grouped separately because it was a pandemic, and we see that in 2050, hopefully it will disappear. Now, in addition to the global burden of disease that is always published by IHME, WHO also works very hard to come up with this priority list of diseases. Here I'm showing you an example of the 17 priority pathogens by the WHO. So these are basically the 17 pathogens that have high burden in different regions. I know it's very hard to see over here, so I'm going to zoom in over here. The 17 pathogens are categorized into 3 different groups. Pathogens that cause diseases where vaccine research, so any intervention research, is very, very needed right now. Then there's another group of pathogens where vaccines are kind of in the pipeline, but a lot more research is required for vaccines to be developed and taken to people. And finally, the third group of diseases, dengue is there, very endemic in Singapore too. There are vaccines that are already, towards the end of the pipeline, about to be introduced in different countries. However, we still need policy regulations, approvals, and also good mechanisms to understand the impact, the biological impact of these vaccines. Now, these priority list of pathogens or diseases are extremely hard to come up with. WHO has dedicated groups who work for years with experts all around the world to figure out what should be the priority diseases that funders, that researchers should focus on in the next decade or even more. I am showing you an example, and the publication is right there. So if you're interested in understanding how WHO comes up with priorities, all the details are in this publication. Because I come from microbiology background, I'm showing you the example of pathogen priority list. However, WHO comes up with priority lists for all diseases, including non-communicable diseases like heart disease, mental health, etc. So something to definitely keep in mind. I'm going to spend the next 4 minutes I have talking about our work in Bangladesh. So in Bangladesh, we started single-cell genomics in 2024. So that the first single-cell run in Bangladesh happened in January 2024. And we are very, very proud to say that we do single-cell genomics in the community, with the community, and for the community. Everything is done in Bangladesh. So we co-design all our projects with the community, as you can see in the first picture. All our sample collections, data collections are done by our community, people trained from that same community. Even when the project leaves, the people with their expertise stay. And finally, the work is done in a lab in Bangladesh. The work that I'll be showing you now is co-led by Dr. Yogesh Huda, who is a senior scientist at CHRF, unfortunately not here this time. So one of the projects that we started with is basically contributing to the CZI Pediatric Cell Atlas. And over here, we looked at the impact of respiratory syncytial virus, or RSV, infections in children under the age of 2 years. So RSV causes lower respiratory tract infection, can cause severe infections, and it's most severe in countries like Bangladesh, India, Pakistan, and some parts of Africa. We really don't understand why in many parts of the world RSV causes mild disease, but in our parts of the world, RSV causes very severe disease. Why are there so many deaths and hospitalization due to RSV? Is there a difference in pathobiology? What makes a mild disease severe? In order to understand that, we started enrolling children that have an RSV infection, and then enrolled healthy age, neighborhood matched, sex-matched controls. We just started sequencing the cells. So far, we have sequenced about 23,000 cells. There's a UMAP here. This is in collaboration with Jose's lab in Harvard and his team. If you want to know more details about this project, please go visit Priyanath and Deb on there and see their posters 70 and 71. We're already starting to see differences between healthy cases and cases with RSV infection. There are definitely different pathobiology that we are picking out. The next project that we're currently working on now is basically understanding what role does gestational age play on immunity, our development of immunity to vaccines in children. So 20% of all babies that are born in Bangladesh are preterm, which means they're born before their gestational age is more than 37 weeks. So they're very tiny, they're not fully mature, they're premature, and it's a huge cause of neonatal mortality. Bangladesh has very high vaccination rates, so more than 95% of our babies and children are vaccinated, and these premature babies go into the same schedule of vaccination. So if a mature baby gets a vaccination at 6 weeks and 10 weeks, the premature baby will do also. However, now we know that premature babies cannot develop the same kind of immune response after vaccination as mature babies. We do not exactly understand why. And should we work with our policymakers to perhaps optimize the schedule of the vaccine so that they develop better immunity? And for that, we really need to understand what are the differences in immune responses in premature babies and mature babies. So we started enrolling 100 pregnant women. So far, we have enrolled 1,000 pregnant women. We start communicating with them in their first trimester. They do their ultrasound with us. And finally, we follow them through their entire pregnancy, so we know the exact gestational age. And after the baby is born, we are collecting longitudinal samples at different age points and after different vaccinations or before different vaccinations of the baby. So our plan is to collect 800 longitudinal samples from 100 term babies and 100 preterm babies. So hot off the press, We just loaded the first 3 samples last week into our sequencer. Of course, too early to say anything, but Opurbo in poster 43 can tell you more about what we are finding and what our plans are. This is in collaboration with Alex Shalak and team and funded by the Gates Foundation. I'm going to end there and end by saying that it's really, really important that we map diseases in addition to health. It's important that the science we do is centered around people and not just data. And we must always remember that science must be done in service of humanity. Thank you. I will take one question. Maybe there aren't. Oh, yes, I see a question. Thank God. Can we get a mic there, please?
So by the way, when I did my master's, I got the lowest grade in epidemiology, but I found it to be, no, I mean, it's epidemiology, but on a computational epidemiology, I did well. So just to clarify, in epidemiology, we always account for potential confounders, right? And I wonder about the statistics that you mentioned earlier, where diarrhea is less likely to happen worldwide as compared to encountering an injury or a road accident, something like that. Were there potential confounders accounted for there? Because I feel like here in Singapore, it's very less likely to encounter an injury as compared to other places.
Thank you for that question. So if I understand, how did diarrhea make it to the top 10 list compared to some of the other diseases like cancer, etc.?
I mean, to account how did potential biases were accounted in terms of ranking those, because I think if we apply that to Singapore, although that is global, right? If we apply that to Singapore, I think the ranking would be very different. If we apply that to the Philippines, ranking would be different as well.
You are absolutely right. So this is global burden of diseases, right? And about 80% of the world's population live in countries where some of the burden of these diseases is very high. Diarrhea is extremely common in many, many countries of Africa and many countries in Asia. And diarrhea happens very early in life. So diarrhea can lead to death. And diarrhea doesn't happen, due to one virus or one bacteria. A combination of viruses and bacteria can lead to diarrhea. And also what happens after a diarrheal episode is it has been shown that children cannot mount immune responses as well to different vaccines, so they're more likely to die from other infectious diseases also. So the DALY due to diarrhea, because it happens so early in life, that it's really high, and it can also lead to different kind of illness, to stay at home, miss school, etc. However, there are many good vaccines for diarrhea now. Most of the world has already incorporated, for example, the rotavirus vaccine. And you will notice that by 2050, diarrhea is no longer on the top 10. It really goes down. So it is decreasing. Thank you. We are going to move on to our next speakers now, are the speakers on the front row. Okay, so we're going to start with Chris, and Chris is going to talk about cerebral malaria.
Perfect, thanks. Hi, so I'm Chris Moxon. I'll wait for my slides, but I can introduce myself without my slides, hopefully. So I'm a senior clinical lecturer at the University of Glasgow, but while my home institution is Glasgow, I actually live and work in Malawi and have done on and off for the past 17 years. Let me get this. So really grateful to be invited here. Fantastic to meet this extremely generous community and meet many people I haven't met before. So thank you very much for the invitation. And it's come up lots here, but while as a clinician, I'm extremely excited and can clearly see the value in a healthy cell atlas. what really excites me about this project and this community and its potential is the potential for translation. And I'm going to start by saying that nowhere is this potential more important than on the African continent. So this is, of course, because of the massive disease burden. 70% of infectious disease deaths occur on the African continent. And this has a disproportionate burden on children. We've heard about DALIs, we've heard about early childhood mortality. So nowhere is this more prevalent than the African continent. You can see that very clearly in these maps. We also, there's a fantastic focus on diversity. So many, beautiful efforts and really impressive things come out of it. Of course, diversity has originated from the African continent, where humans were originated, and which is still probably the most ethnically diverse continent. But I also want to say that whilst it's extremely variant because of genetics, genetics is not the only source of variation. We're thinking about cells and cellular responses. And these don't just become because of genetics, they become because of environmental influences. We heard about the influence of diet earlier in a wonderful talk from Yusuf. But also infection is a huge influence on the phenotype of cells and on their responses. And of course, if you look at the burden in the upper left corner, you recognize that on the African continent, this is going to have a huge influence on cells and their responses. And just one demonstration of this is our paper for the same virus, SARS-CoV-2, whilst the clinical phenotype and histology has similarities, we realize that this is superficial. If you look at the cellular response in the lung and the molecular responses, they're really very different. And of course, some of this work, comparison with other countries, was only possible because of the human lung cell atlas, so already demonstrating the huge importance of these initiatives. So hopefully I've convinced you thoroughly that it's really important to be doing this sort of research on the African continent. But not everybody's convinced, and there's a huge mismatch between where there's importance and need and value. Value because I say if we've got all of this genetic diversity or this phenotypic diversity, then many of the answers, diversity, heterogeneity is a huge source of biological understanding. So many of the answers to questions for disease that affect people across the world may well be found on the African continent, but the investment is much lower, and this is just compared with GDP. So these are poor countries, and still the investment compared with GDP is far lower than most other places. And I want to mention, doing work on the African continent, that this, of course, is a brick problem because it's much more expensive, an order of magnitude more expensive to do the research in this context. The last thing to say is that the population is growing, and by 2050, when we had projections just now, a third of the world will be, under 25, will be in Africa. So this is a bit about opportunities and barriers. So I live in Malawi and work in Malawi. There's great opportunities here. It's a beautiful country, but it's very poor, high disease burdens, particularly infections. We work in a campus that has a central hospital, Queen Elizabeth Central Hospital, massive referral center for a big part of the region. We're linked with a scientific community, with MLW where I work, Blantyre Malaria Project where I work, where we have really good scientific facilities from Wellcome, but also, previously NIH. Who knows what happened with that. So, we've managed to set up many things, other people previously, and then more recently we've set up single-cell sequencing. Many challenges, as I've highlighted. Some of our single-cell sequencing work and our ability to characterize disease depends on autopsy studies. There are historical autopsy studies, and more recently we've set up minimally invasive autopsy platform. This is dependent on really hard work from pathologist Steve Camisa, who I have a long collaboration with, and with social science and medical ethicists to make all of this sensitive work possible. We've done a ton of sensitization work. So I'm going to talk to you about cerebral malaria. You've heard about the complex malaria life cycle. Cerebral malaria is called by Plasmodium falciparum. It causes almost 200 million infectious episodes on the African continent every year. You did hear that right, 200 million. And that burden is really difficult to imagine, but causes huge amounts of suffering and about 500,000 deaths. Cerebral malaria is the most severe complication. Despite highly effective anti-malarial drugs, about 20% of children die, and about 50% have either neurodisability or developmental complications. We don't yet have a adjunctive treatment because it's really difficult to understand the processes that lead to this very complex disease of coma and seizures. Seminal work by my colleagues in Malawi using an MRI study established that brain swelling is a critical cause of death. The brain is in a closed box, and so if it swells, there's nowhere for it to go. This compresses the respiratory centers at the base of the brain, and you stop breathing. So this is an opportunity to understand how we might treat it. Work done on the retina of the eye, visualizing the neurovasculature, and histology work that we and others have done has established that blood-brain barrier breakdown is critical for this swelling, and that this occurs at sites where the infected red blood cells stick to blood vessels in the brain. This is an opportunity. If we can understand how infected blood cells sticking to the blood vessels cause this leak, we have the basis for an intervention. When children present, they're on a knife edge. We give them treatment, and some of them improve, actually often quite quickly, but others either deteriorate and die or have complications. And if we can reverse this rapid swelling and blood-brain barrier breakdown, we can improve outcomes. We consider that the parasite sequestering can cause this blood-brain barrier breakdown through two main concepts. One, indirect interactions with immune cells that then cause the damage, and the other through direct interactions of the parasite. And we're using different systems biology approaches to understand immune responses, and then spatial approaches in post-mortem tissue to understand these two potential mechanisms. And I'm going to focus today on some work on the periphery on neutrophils. Work from lots of people has established that neutrophils are probably really important in the pathogenesis, including a type of neutrophil activation called neutrophil extracellular traps, NETs. This is what a net looks like, and I don't know if somebody's able to, so NETs are where the neutrophil explodes and extrudes DNA. And you can see this process in action here. We use a DNA dye that doesn't get into the cells, that's the purple, and then the green that does get in, that's purple, and then the green that's outside. You can see the neutrophils exploding. And you can only imagine what this, with the DNA, toxic granule proteins, and histones, does to your vasculature. And in many inflammatory disorders, this is a critical source of, and cancers is a critical source of vascular damage. So we looked whether there were nets in the brain. We used an antibody provided by our collaborator, Borkato Mulik, that's highly specific for nets. It recognizes a clipped form but in late nets. And those are in green, and we've scored this. So hold on. We've scored this, and we can see that there's more nets. I'll show you that in a minute. But we also really want to see that these are true, that this really are identifying nets and not some artifact. And so we used CLEM, correlative light electron microscopy, and you can see that the green superimposes on this. I don't know whether the laser works. Superimposes on this clear net structure here of extruded DNA that's poking through an area of blood-brain barrier breakdown. And we've scored this using an automated computer pipeline, and there's much more nets in the brain in the cerebral malaria cases. So then going into the periphery to understand how these neutrophils might be different and why you're getting so many more nets in the cerebral malaria cases. Firstly, we looked at nets in the blood, in the plasma, in different clinical groups. Healthy controls, children with other causes of coma, so non-CM, children with malaria in the periphery, but where they don't have pathognomonic retinal changes, a group called retinopathy negative, that's a great control. They don't have sequestration in the blood. We've shown that post-mortem. And so they represent probably a different mechanism of disease. So it's the retinopathy positives are classic cerebral malaria that have these nets. And there's no more neutrophils in the blood compared with the other really sick groups. But there are much more of these low-density neutrophils, a group of neutrophils that separates off in the buffy coat when you do a gradient separation. And then we did a functional assay. So this is nets in the plasma, but this is a functional assay testing the properties of the neutrophils and using different stimulants, PMA and a calcium ionophore. And you can see the calcium ionophore induces much more nets in the low-density neutrophils, suggesting calcium influx induces much more, that these are much more net-prone neutrophils, at least to calcium influx. Calcium influx is a really important mechanism of mitochondrial ROS and the way of inducing nets, so they're different net mechanisms. So we looked at the neutrophils, stained them and did confocal microscopy on these, and malaria neutrophils are bigger and longer than those from controls. And then we separated the neutrophils out using negative magnetic separation and then divided them into the low density and the normal density and then did bulk sequencing on the two. We recognized a whole load of net proteins in this volcano plot, but also mitochondrial proteins came up in a number of the different pathway analyses. And then to try and understand the origin of these abnormal net-prone neutrophils, we did single-cell RNA sequencing on neutrophils, both separated and from whole blood. I'm going to rush through this a bit because of time. We also took cells from the bone marrow from post-mortem cases and from some samples obtained in orthopedic operations. And we were able to do a construction of using pseudotime of neutrophil development. And in contrast to some previous studies that have shown that neutrophils have a very linear development without any subsets developing, we recognize these subsets at the top, a branch point, which leads to a population which is much more commonly or much enriched in low densities, those green ones at the top, so we think that early development in where you've got a lot of sequestered parasites in the bone marrow, early changes are inducing different neutrophil production, and we're trying to do some investigations on that with our colleague Borko using a stem cell-derived granulocytes. Is this just a generic response to infection? We think that there are, of course, very conserved functions. It's an innate immune cell. But we also think there's marked differences, and by comparing neutrophils from cerebral malaria with COVID neutrophils, we noticed that there's similarities. There's also quite a lot of differences. And one of them is that if we look overall in interferon responses, actually interferon responses are down in the cerebral malaria neutrophils, whereas they're way up in COVID. But then if you look early on in the neutrophil trajectory and earlier neutrophil populations, actually the balance is the other way. So there seems to be changes that occur that are dependent on the maturity state of the neutrophils. Does this have functional consequences? Well, blocking a mitochondrial protein, mitochondrial calcium uniporter using rhodidium red attenuates net response. So this is the unstimulated to nets, you can clearly see them here, and then when you treat with the compound, you get almost completely lose any net formation. So just putting this together, as I said, we think that parasite events in the bone marrow or parasite sequestration in the bone marrow affects neutrophil development, leading to a net-prone form of neutrophils that then, when they are exposed to heme, with previous publication, Borko's publication that we collaborated on, that shows that heme from rupturing infected red blood cells promotes net formation. Still plenty of questions and more work to do, but I think this is really exciting. A sneak peek I wanted to try and present some of this stuff, but there's no time, so maybe you'll have to have me back again. But we've been doing work on the brain, and we've done some geomics and some cosmics on a 6,000-plex platform to try and understand what are the mechanisms in the brain and endothelial cells and other cells, and can see some really nice patterns around the areas of blood-brain barrier breakdown, currently mapped 200,000 cells. So a ton of people involved in this work from other labs across the world, a ton of people involved in Malawi and sensitization, the clinical side, setting up the research ward. So these are my teams in Glasgow and Malawi, but also completely indebted to the foundations of the research ward, which started well before I arrived. The late Malcolm Molyneux, Terry Taylor, and Carl Seidel, and then from my colleagues, Yamakani, a pediatrician that I work with, and Steve Kamisa. So thank you all very much.
Thank you, Chris, for that wonderful talk. Sorry, we don't have time for questions, but I'm sure we will find you in the next break. We will quickly move on to the next speaker, Zhang from Shenzhen Bay Laboratory.
Thank you for the invitation and the introduction. I'm Zhang from the Shenzhen Bay Lab. I established my own lab in 2021. My lab mainly focuses on understanding how immune cell heterogeneity influence the consequences of immunotherapies, in the gastrointestinal diseases, including colorectal cancer, which I have done during my postdoc work at Professor Zhang's lab at Peking University, and the liver cancer that conducted in my own lab. So today I'm going to mainly talk about the T-cell dynamics in the liver cancer study. As we have known that immunotherapy participates reinvigorating anti-tumor T-cell activity by immune checkpoint blockade has transformed cancer treatment. However, cancer patients show different treatment responses. In colorectal cancer, for example, the microsatellite unstable patients show favorable treatment responses. However, the large majority of these patients that are microsatellite stable patients, they still cannot benefit from such treatment. Another example is liver cancer, that the treatment efficacy is also low. Beyond T-cell-based immunotherapies, there is great interest in treatment strategies targeting other cells and components in the tumor microenvironment, such as myeloid cells. And these strategies, including CS1R blockade that target tumor-associated macrophages and the CD40 agonist that for dendritic cells. We have previously demonstrated the mechanisms of these two myeloid-targeted therapies. Beyond immune cells, recent clinical trials also showed promising results for other therapies, such as anti-angiogenesis. One of the examples is combined PD-1 and lenvatinib. This is the multi-TKI inhibitor. And in a phase 2 liver cancer study, the combination therapy showed improved treatment efficacy combined to PD-1 alone, but the mechanism were not clear. So our question was, what are the immunological mechanisms behind response and resistance to such therapies? So to answer this question, we had collaborated with immunologist Professor Chen Dong at Westlake University and Professor Jinyue Liu from Tier 1 Hospital, who conducted a phase 2 clinical trial in liver cancer. These samples of these patients were collected before and after the PD-1 and lenvatinib combination therapy. So I will go over some of the key findings of CD8 T cells, for example. As we can see that granzyme K positive effector memory T cells, a lot of speakers had mentioned, those are the purple dots. They were increased in the post-treatment responders in the responders. And the green dots, those are the exhaust T cells. They were considered loose effector functions. They show decreased proportions. Additionally, we also observed the large clones of the granzyme K positive effector T cells, indicate they could be activated during the treatment. Since we have observed the compositional changes of subsets of CD8 T cells, so we asked what this could be caused by phenotypic conversion or called state transition. So to answer this question, we used the T cell receptor-based analysis, a framework Star Trek we developed previously. So based on the TCR analysis, we observed that granzyme K positive effector T cells in the heat map in the middle column, they showed TCR shared with both terminal exhausted T cells and effector T cells. So we divided these granzyme K positive T cells into two populations. One shared TCRs with terminal exhausted T cells. They are called the precursor T cells, short for TPEX. So consistent with the notions in this field, these precursor exhausted T cells, they indeed increased in the post-treatment tumors of responders. Interestingly, we also find that the effector memory T cells, that they did not share TCRs with exhausted T cells, they also elevated in the responders. And interestingly, they also showed strong TCR shares with those effector in the blood, indicate that these cells could be circulate between the tumor and the blood. So we named these cells as circulating effector memory T cells, short for CTEMs. So we asked how about those responses in monotherapies? Since our study is based on a single-arm clinical trial, we did not have the monotherapy data, so we combined our combination therapy with anti-PD-1 monotherapy data from Mariam's lab at Mount Sinai. And we can see that after the integration, the TPEX cells indeed has higher proportion in the responders compared to non-responders. But we did not observe the similar changes for CTEM cells, indicate that they could be the reason why the combination therapy showed higher treatment efficacy. So we asked how this could be happened. To answer the question, we have gone back to the preclinical data for the anti-angiogenesis treatment. Before the treatment, the vascular network in the microenvironment is abnormal, which caused an immunosuppressive microenvironment. But after therapies such as anti-VEGFR, the vascular will become normalized and the microenvironment shift to immunosupportive one. So we compared our biochemistry data with those from anti-PD-1 monotherapy and observed that we indeed observed elevated vascular normalization in the combination group compared to monotherapy group. And this accompanied with enhanced CD8 T cell infiltration. Another key question for this study is that how about HPV-specific T cells contribute to the therapy? Because one of the key features of liver cancer in Asia is that over 85% of these patients are linked to HBV infection. And those HBV-specific T cells could be related to immune response. To answer this question, we have carried out the dextramer-based single-cell transcriptome analysis. And for such data, we have developed an analytical tool called DexTrack to quantitatively identify TCR and peptide pairs. By successfully identifying HBV-specific T cells, we found that these cells were more enriched or called captured in the responders. And these cells were more enriched in the circulating effector memory T cells, CTEM cells, as we have talked before. And these cells also showed higher expansion and migration potentials than the other CD8 T cells. And for the analysis, exact signature score revealed that these HBV-specific T cells showed elevated proportion in the responders. So take a summary of the response part. We found that the circulating effector memory T cells enriched with HBV specificity contribute to the improved treatment efficacy of the combination therapy. So next question is, what are the resistant mechanisms? For this part, I will take the KIR CD8 T cells as examples, and these are the major findings in this study. Actually, we have identified two KIR positive CD8 T cells. These cells show the molecular expression similar to natural killer T cell, NK cells. For the subsite enriched in the blood, they responded to those identified in the infection and the autoimmune disease published by McDavis lab at Stanford. But for the tumor CD8 KIR populations, they show distinct transcriptome features. And interestingly, this population also enriched in the non-responders after therapy, but we did not observe such changes for anti-PD-1 monotherapy. So this could be the reason why some of the patients cannot benefit from the combination therapy. And this tumor KIR population also show distinct features in the post-treatment tumors. One of the examples is the higher expression of TGF-B1, which can promote the suppressive function of CD8 T cells. So these are all about the CD8 populations. And beyond this, we also identified key populations that are the CD4 regulatory T cells for the resistant mechanisms. Given this is the Human Cell Atlas meeting, so at last I will give talk a little more about what we have done in this specific scope. So beyond GI cancer, our lab also interested in inflammatory bowel disease. So thanks to the fantastic, excellent work in this field, we began to understand this disease by integrating this published data set, published around 2021 when I established my lab. So after integrating these single-cell transcriptomes from over 400 samples, we systematically compared two subsets of the IBD, ulcerative colitis and Crohn's disease. We identified immune cells and non-immune cells showed distinct enrichment. And we also tried to link GWAS information to the different cell subsets. And we also established a web tour for someone who are interested. Actually, I have to say that the industry actually pay more attention to our website. So at last, I would like to give my special thanks for my collaborator, Dr. Chen Dong from Westlake University, and the clinician, Jinyue Liu, and my postdoc, Dr. Hu Nie, and Dr. Jason Lee, who did the analysis. Here are my founding supports. Thank you.
Thank you. Thank you so much for that wonderful talk. We do have some time for questions. Yes, Zhang. We hear you.
Actually, it's not my slides now.
Can you repeat the question, Zhang? We cannot hear you, actually.
What was the effector T cells into the tumor?
You mean the liver cancer study? Yeah, this actually they are the subpopulation from the Gram-K positive T cells. We define this population by their TCRs. They did not share TCR with those exhausted T cells. And interestingly, what we saw this were interested because they share TCRs with those in the blood. I guess you have observed this actually in the large blood data from use. Yet, and another point is that they correlated to the favorable response in the responders. Yeah, and several remaining questions actually includes what they did recognize for the antigens. We haven't finished that work. Yeah. Thank you.
Thank you. Thank you so much. We are going to move on to our next speaker, Jong Kim from Seoul.
Okay, hello. My name is Jong-Il Kim from Genomic Medicine Institute at Seoul National University in Korea. I'm very happy to introduce our SCATE project in this meeting. Today I cover the SCATE introduction and three unpublished disease data from 3 organs and some integration issues. So what's SCATE? SCATE stands for the Single Cell Atlas of Immune Disease. And the logo is designed by Dr. Seon Lee from our lab. It symbolizes the two cells, the immune cell and parenchymal cells and their interaction. And the color set is from dancheong, is the traditional multicolored paintwork found in Korean palace rooftops. So the key concept of SCATE is Korea and immune system and disease. As Sarah mentioned yesterday, the SCATE is a part of the ongoing Human Disease Atlas initiatives. And as you can see in the slide, our SCATE is perfectly aligned with the next phase of the HCA because they aims to more diseases and geographic and ethnic diversity. So the SCATE project is the Korean government-funded top-down project for some flagship atlas in Korea, and they supported about $16 billion over 6 years until 2027, and about more than 100 researchers, immunologists, clinicians, and biostatisticians are participating in our project. Although about more than 30 PIs are joining this project, these three are the key persons. Hyun-jae Kim is dermatologist, and Jung-Seok Lee is a rheumatologist. They are clinicians, and they are expert in immunology and genomics. So these two PI produced all the samples, data for our project. And Jong Park, he was the previous HCA member until he joined the KAIST, and he is doing a critical role in designing and integrating our data set. So this is the list of the initial list of the 16 diseases, our target disease and the selection criteria. And this is the scheme for sampling and profiling. And the map is for the geographic advantage of our project. So last year we published a short comment in Nature for this kind of small regional consortium. So this is the original pipeline we suggested when we started our project. So in the first half, 3 years, we will focus on data production, and we'll open out the first version of our atlas and we did. And we'll make the final version in 2027. But when we compare our standard to the level of the HCA, we should admit that our first version must be 0.5, and the final version 1.0 will be available in 2027. So from now on, we'll introduce 3 different projects we did. First one is the myocarditis. This project was done by Jinyue Liu from KAIST and some cardiologists. So the myocarditis become very familiar because is the distinct adverse event specifically associated with the mRNA COVID vaccines. So there are 2 kinds of myocarditis. Conventional myocarditis is before the COVID-19 era. So we don't know the exact pathogenesis, but most in cases is uncompleted, but some has very high mortality risk. So there is some evidence that emergence hematopoiesis and CCR2 axis is important in the mouse model. And another category is the vaccine myocarditis. So compared to the conventional myocarditis, there is a low instance of adverse clinical outcomes and young male dominant, and it may need medical management for several months. So it applies that there will be some potential difference in the pathogenesis mechanism. So, and there is some issue that the pre-spike antigenemia is found. The antigen is increased in blood when the patient developed the vaccine myocarditis. So this is study design. They selected the pre-COVID conventional myocarditis patient and vaccine myocarditis patient, 6 and 6. They collected sample in acute and recovery phases. And for the control, they used the normal vaccinated control without any symptom before vaccination and 1 week after vaccination, and the 1 week after first dose and 1 week after second dose. And from one patient, they got the sample from the heart tissue, the heart transplantation. So this is the tissue and blood data, and they found that cis-FAL2 positive classical monocyte, the myocarditis specific, and so in the blood and tissue, this cell is very specific, this specific pathologic subset. And okay, I should see this one. Sorry. So when they compared the BAM, vaccine myocarditis, and conventional myocarditis, they found that the vaccine myocarditis BAM exhibit more inflammasome-driven signature, while COM is dominated by the type 1 interferon responses. And when they did some experiment, so they applied the spike protein and LNP, and they found that you can see the spike protein alone induces some the inflammasome reactions, but when it is combined with the spike protein and LNP, it shows the additive effect. So that means that so the LNP, the high spike protein and LNP combination may cause this inflammasome activation, and they showed also pyroptosis and the monocyte. And for the T cell, they showed some pathologic TH17 signature in BAM, and CXCR positive, CXCR2, CXCR16 mediated activation in BAM. This is the unique feature of their study. They did TCR sequencing, so they for the T-cell clones, they could identify the shared clones. So and check compare the acute and recovery phase. When they use the old T-cell subset, those T-cells, there's no difference. But when they focus only the shared groups, they found some features. So I skip this. So this is the summary. So both diseases showed inflammasome, type 1 TNF and iron beta signature and interferon signature at acute phase. But for the recovery phase, the BAM has consistent TNF iron signature activation, but the interferon signature go down. And COM has the consistent interferon signature activation, but the TNF and iron beta signatures goes down. So that's the switching off of the one system. Maybe it is the difference between the two diseases. So this is summary. So they discovered myocardial-specific CCR positive classical monocyte in the patient blood and heart tissue. And they found that the COM is related to interferonopathy, and BAM is inflammatory, but acute phase, they two coexist. And it's the epidemiologic study showed that the higher myocarditis risk is for second dosage, and there is low risk when in the delayed subgroup. So they suggested 4 weeks interval, but if it's delayed more than 6 weeks, there's definitely the low risk in the myocarditis. That may be explained by the spike protein, the additive action of spike protein RMP. And there is some suggestion for using the immunosuppressive agent for the treatment. The second topic is in ILD. So it's directed by Dr. Seun Lee and collaboration with Dr. Jong-Il Lee and Hyunjae Kim and cardiovascular surgeon and rheumatologist. So there's many study about the idiopathic pulmonary fibrosis, but we focused the ILD associated with the systemic inflammation, connective diseases. So we used the three strategies. So proper atlasing and utilizing the pre-made reference atlases and maximizing code design. So we try to follow the proper atlasing guideline. And this is the scheme. So from 17 donors, we collected 12 donors. We collected 11 PBM sample and 24 tissue samples. Some tissue samples were acquired from the same patient, in the several regions with different severity. And we did hierarchical cell type annotation, same to the human lung cell atlas. And this is the scheme. So for the tissue data, we used the human lung cell atlas as a reference. And for the PBMs data, we used the IDA data, especially the Korean IDA data as a reference. We checked the integration benchmark that the integration was properly done. So using this SGRC and the human lung cell references, we did the reference mapping and the uncertainty, and also we used the normal lung data, not in HCA, and some pulmonary fibrosis data as positive and negative controls. Using this uncertainty data, so we can see there is a divergence from our sample to the references. And this first one, the previous known finding is this one. For the many pulmonary fibrosis disease, three different cell types was already identified doing the key roles. This aberrant basaloid and myofibroblast and the pro-fibrotic macrophages. These three cell types are already known. But when we did analyze our data, first in the stromal cells, the mesothelial fibroblast is the highest difference, shows the highest difference, as well as the myofibrotic macrophage, myofibroblast. And from the left side, among these epithelial cells, in addition to aberrant basaloid, already known, we found that ICAM1 positive, 81 and 82 has some different features from references, the control data. And the right side, for the macrophages, so new cluster shown in cluster 7 is MMP19 positive macrophage subset, and the gene set shows that is the gene programs related with the foam cell differentiation, very similar to atherosclerosis in heart disease. So we named this foam cell macrophages. So this is a summary. So in addition to previously known 3 cell types, 4 different additional cell types, 81, ICAM1 positive, 81 and 82, and the mesothelial fibroblast, and foamy macrophages are doing some essential roles, probably in response to the systemic inflammation. And we also did analyze the PBMC data using the IDA as data as control. And when we did make the trajectory from healthy to the very severe patient, we found that there is a tendency that some naive T cells and CD4 helper T cells decreases, and NKG2C cell, NK cell, cytotoxic, and cytotoxic CD8 T cell is increasing by the severe progression. And I'll skip this one. So this is summary. We built a comprehensive ctDNA ILD single cell landscape, integrating both tissue and paired PBMC, and single cell level identification shows novel cell status, and systemic immune dysregulation, the cytotoxic and naive imbalances is linked to the local pathology. The third disease is PPP. It's a palmoplantar pustulosis. I think the time is short. So, okay, this is a kind of the psoriasis, but quite different, a little bit different. So I skip most of the data, sorry. Just showing this summary. Okay, this summary. So we found that PPP still shares the key immune players with the conventional psoriasis, particularly TH17 driven immunoinflammation. But the difference in treatment response, it doesn't respond to the conventional treatment for the psoriasis. So the difference in treatment response may not arise from the entirely distinct mechanism or cell type, but rather from the composition of fine scale immunologic distinction. And we found that, so the single cell data, we did single cell and the Bijum HD, so this is the Bijum HD data. So in both data, confirmed that some densely organized CCL19 positive dermal lymphoid architecture. So this fibroblast is similar to the lymphoid organs. This is prominent environment of the dendritic cells and degree of TH17 plasticity and sustained myeloid lymphoid crosstalks. And we also could suggest some potential target drugs from our data. So there are both research data on our scale. There is poster presentation and oral presentation. So this is the last part. So until so far, my presentation is focusing on the diseases, not atlas. This is because in first phase we had to focus on the data production, and many researchers are involved. They have some pressure for publication, quick publication. So we should focus on these one by one. But now it's in the mid phase of project, so we are shifting our focus to more atlas integration and the data, getting the insight from the combined data sets. So that's the first our intention data. So we made integration about 2 million cells, from 300 donors. And we try to use our data for some integration. This is one example. This is Dr. Jong Park's work. He made the Pangea project. He used the public geodata with the FastQ. So with the data, so he downloaded the web crawl, all the data, and did alignment again. And he removed some data, and finally he got some more than 12 million cells. And he then combined the curated data set without FastQ. So he could make good atlases. So he selected 10 cell types across all the tissues, and did the hierarchical cell type annotation, manual annotation. And with this data, he tried to use some, get some insight. This is blood sample data, about 1,000. He used the cellular composition as pictures. So the one dot in this data is just one sample, but the cellular composition is the features. So when he did the clustering, he showed that 10 different clusters. So blood phenotype was shown, and some BP, there are some abundance, the disease sample abundance.
May I please ask you to summarize quickly?
Sorry, okay. So this BP program is related with some diseases and some gene programs. And we tried to this data set, our scale data set, and use our blood data, and found that the all data, it is clustered with 2 cell types. One is the CD4 activated, and one is the CD8 dominated. And interestingly, for example, liver disease, the NASH is the upper, the HD4, and NAFLD is the CD8 positive. And even in MS, multiple sclerosis, there are the disease classified 2 different subsets. Interestingly, the one set is...
Sorry, we really need to wrap up.
Sorry, okay. It's a related response. So that's the last slide, sorry. Yeah, okay. So I like to thank you, my colleague, for the OHS.
Thank you. No, thank you. Thank you so much for that wonderful talk. Sorry, we have no time for questions. We're going to move on to our last but not the least speaker, Jinyue Liu from Genome Institute of Singapore. Thank you.
Hi, you hear me okay? I don't think you can see me, but you hear me. Okay. Okay, I'll try and do this swiftly, not too quickly, but swiftly. Hi, so my name is Jinyue, and I'm from the Genome Institute of Singapore. And my lab is interested in understanding the processes that regulate the birth and death of cells in the human brain. And today I'll be sharing with you some of our unpublished work using autism spectrum disorder as a case study. Now, almost everyone here in this room would have met someone who has autism. And what is striking is that the person may present symptoms that appear very different from someone else with the exact same diagnosis. And this is what we describe as clinical heterogeneity in autism spectrum disorder, and it's one of the, I would say, biggest mysteries about this condition. And it's a neurodevelopmental condition, and more than 1,000 genes have been associated with this condition. So in general, the field has been trying to propose several hypotheses to understand the mechanisms underlying autism. And one of it that has been proposed is what we call molecular convergence, in which there are the genes that are involved in autism may perhaps participate in common pathways that are shared across different patients. And there are multiple evidence that point towards this hypothesis. For example, there's abnormal regional patterning in the cortex across different patients. And also you see different risk factors enriched across different developmental time points, in particular during the early to mid fetal gestation weeks, as well as upon birth. And also some work showing the enrichment of differentially acetylated peaks upon birth. Now, what is less known about autism is that in the brains from children with autism, there are actually some cellular pathology. So what you're looking at here is for in situ hybridization images of 3 different genes. So each color represents a different gene. And what you're seeing here is a hole where the expression of these genes are missing. So children with ASD, autism spectrum disorder, actually present these focal patches of cortical disorganization, and they vary in the layers and the cell types that are affected. So on your right, you will see that there's quite some heterogeneous gene expression across the panel of genes that were assayed, and this is indeed the case. They are quite heterogeneous. Now, we were interested to understand a little more about how the autism spectrum disorder might occur. But one of the tricky things is that its pathogenesis actually begins in utero, but the diagnosis is postnatal. And so what we really have is to model these condition using organoids, brain organoids. And so we are not the first ones to do it. There have been multiple foundational work that has been done in this area. And people have identified excitation inhibition imbalances, asynchronous neuronal development, and several other cell type proportion changes. And they are very often dependent on the genomic context of the individual. And so, but what is not yet understood or not yet investigated is how the various cellular changes may contribute to the tissue architecture in these patients. And so here's our study design. We have controls and patients. And because I get this question a lot, I'm just going to answer it right now. Our patient pool is very heterogeneous. They comprise of both idiopathic as well as genetic forms of autism. And we get induced pluripotent stem cells from these patients, and we generate cortical organoids. And we perform both single-cell RNA sequencing as well as spatial transcriptomics. And in this case, we are using MERFISH. And we look for a certain set of phenotypes, such as cell states, spatial organization, as well as their cell-cell interactions. Now, what you're looking at here is a brain organoid in our hands. You'll see those little buds on the surface. You can think of each of those buds as a unit of the cortex. It's what we call neuroepithelial buds. And you can take cross-sections through the organoid, and this is what it looks like with MERFISH. So this is assaying for 500 plus genes, even though I'm showing you just 6 genes. Even then, you can already see pretty nice layering, cellular layering with just 6 genes, marking the different types of neurons and progenitors in the system. Now, if you look at the autism organoid, you would first off notice that there's some disorganization and disarray going on, even from just these 6 markers. Now, if we take a closer look at perhaps the neurons, because we identified, us and many other labs have identified neurons as a key locus of pathology for autism, you'll see that there are some interesting phenotypes there. So first off, what you're looking at here are spatial metagenes obtained using MERFISH. So blue and red are represent 2 different classes of neurons. And in the control, you see that they are very nicely layered, or one on top of the other. Now, if you look at the autism organoids, here I'm showing you 3 different samples, you'll see that they are completely disorganized. And we can quantify this using an index called lamination index. Now, this is very exciting to us. Now, if you look at the spread of values in their lamination index, you'll notice that it's not all or none. And so we were wondering, perhaps there are different ways in which the neurons are becoming disorganized. So you can imagine that in the controls, they are perfectly layered, no issues there. Now, in the autism cases, perhaps there is local disorganization of the neurons, or perhaps it's just a global disorganization. So everywhere the neurons are just disorganized. And so we take a closer look at our merfish data, and what we saw is that perhaps it's the local form of neuronal disorganization that's happening. As you can see in these two bars, the first one, number 1, is layered, and the one number 2 is pretty disorganized. And we can quantify the proportion of cortical plate ROYs that are disorganized, and you can see a substantially higher proportion of autism organoids are disorganized. And so importantly, we could validate this disorganization now. And what you're looking at here are protein stains for the 2 neuronal classes. And you can see that they are indeed heavily intermixed in our autism organoids. So on the left, you have the yellow and blue bands representing the 2 different types of neurons. And on the right, you can see the autism organoids showing complete disorganization of these 2 markers. And we can quantify this difference broadly across all of our organoids. Now, with spatial information, as all of you would appreciate, you can actually start to interrogate the cell-cell interactions that could be responsible for the cellular phenotypes that we see. And so we devised a metric called spatial signaling score, where we quantify the interactions between pairs of cells that are in contact with each other. And with this method, we found that, interestingly, even though it's the neurons that are disorganized, it is the progenitors that show the majority of the affected signaling pathways. And a lot of these signaling pathways participate in cell adhesion, for example, extracellular matrix, integrin signaling, and so on. Now, we next asked whether any of these interactions and cellular phenotypes have longer-term consequences, because you could imagine if the pathology begins in utero, but the behavior only shows up later on in life, perhaps the pathology would continue to persist in the patient. And so we started looking at some of the structural and longer-term impact in our organoids. And we assay for radioglia because that is a very important scaffold within the developing human brain. This is the scaffold upon which the neurons migrate on. And we can see that they are using nestin as a marker for the radioglia scaffold. You could see that it's pretty much disarrayed or abolished. And if you look at 30 days or later on in the organoid life, you can see that the disorganization continue to persist within our autism organoids. And so in summary, what we have found is this spatially mosaic pattern of pathogenesis that seem to occur during the prenatal brain development. And we like to think that it might contribute to the heterogeneity that we see in autism spectrum disorder. And we did not cover today, but we also discovered some effects on the developmental timing, concurring with a lot of the literature and the work that has been done so far. And so with that, I'd just like to thank a lot of the major people who have been involved in this work, including Sham, Kok Hao, Hui Kwan, and Lindy, who has done most of the heavy weightlifting here. And with that, thank you.
Amazing. Thank you so much for that wonderful talk. Do we have questions on the floor? Yes, at the back. I think I saw a hand. No, someone was yawning.
If you have questions, you can email me as well. I just realized my email's out there.
Hi, thank you for this amazing talk. I had a question about whether you've observed some correlation between the level of disorganization you see in the organoids and the level of support these individuals need in daily lives.
I didn't catch the second part. Do you mind repeating? Sorry.
Oh, sorry. So you've observed some disorganization in the organoids, and I'm curious if it correlates to the level of support these individuals need on a daily basis.
The level of what?
Like a support from other people, like in terms of like their symptoms.
Oh, okay. Thank you for the question. Yeah, I do get that a lot as well. So your question is how well do the degree of disorganization correlate with their clinical symptoms? Is that right? Yeah. So in this case, because Our samples are commercial, so we don't have access to their full detailed patient metadata, and that is really one of our regrets of the project, I would say. Yeah, but I think it would be really interesting. So if we can say repeat the project with newly recruited patients, right? And we can compare the degree of disorganization in their organoids and their scores, like ADA or ADIR or ADOS scores, which measure their behavioral symptoms. Yeah, I think that would be really interesting.
We have another question in the front.
Okay, so we have not looked into that, but I think that's a really interesting question. So our patients are genetically diverse, that's for sure. We identified different candidate variants that in each patient. Perhaps I can catch you after this to see how we can do this. Thank you.
Wonderful. Thank you so much for that. And that brings an end to our disease session. I believe we break for caffeine now. Thank you all.
Yeah, employees are very